kids is probably experiencing a similar thing, and everyone, regardless of whether you have kids, is experiencing a weird, this is a very strange time. So I want to thank everyone for showing up to the seminar today. We're of course testing out this Zoom format. If this ends up working or not, we will that'll gauge whether we continue doing this. So please email your feedback to me as well as this is happening and unfolding. Reflect on whether we could do a better job. If there's anything you know about Zoom that we don't, because we're learning too. Yeah, so we want to try to maintain some sense of normalcy in the department, and maybe this is just exacerbating it, but we want to, Stephen Fried was open to coming and giving us a seminar. So he is a new member of the CMDB program at Hopkins, by which I mean he is a faculty member in the CMDB program. His home department is chemistry. He did his undergraduate training in chemistry and in physics at MIT. He did his PhD in chemistry at Stanford, where he worked with Stephen Boxer on origins of catalysis in ketose. Intro brief, Stephen has received numerous awards, really impressive awards, including as an undergraduate. He was in the MIT Department of Chemistry, the Alpha Chi Sigma Award. He was inducted into Sigma Pi Sigma, which is the Physics Honor Society, as well as Phi Beta Kappa. He's also attracted significant grant support already very early in his career, including the Phyllis Albstein Foundation for Brain Science Grant, a Johns Hopkins Discover Award, and a Human Frontiers in Science Grant. And so last thing I want to say is that while our presence is virtual, the science is real. So feel free to raise your hands if you can figure it out on Zoom and ask questions. I think chatting might be the best way to reach everyone. So with that, I want to introduce Stephen, and let's see if this works.
Wonderful. Well, first of all, I might want to say, can you hear me?
So maybe people can type a chat if you can hear. I can hear.
Yeah, it sounds good on my end.
Yes, we're getting lots of yeses.
Okay, perfect. That is all I wanted to start with. And also thank you to Will for the invitation and for the very kind introduction. I guess as a bit of context to this, I think we were operating at one point under the assumption that this would happen when no one would be able to travel, but we'd still be able to congregate. And so I think this was originally designed to be done in vivo, as it were, but then with the rules applying to groups of people, we are trying this experiment, and hopefully, I guess I'm an experimentalist, so I like to try new things and see what works, so we'll find out. All right, so I'm a biophysicist, really, even though I'm in a chemistry department, and I'm showing you here a picture of an experiment that biophysicists are very good at doing. It's an experiment that we've been really doing since the 1960s and 70s, when it was pioneered by Christian Anfinsen. Essentially, when biophysicists want to study how proteins fold, what they typically do is they begin with a small, relatively reasonably sized protein, something like RNase H, so 100 amino acids. You do something to get this protein to unfold, which typically involves using chemical denaturants, but can also, if you're particularly fancy, involve using mechanical force as well in a single molecule experiment. And then with this unfolded protein, we do something to get it to return to its native state. So we change the conditions so that it's in a condition where the unfolded state prevails to a condition where the native state prevails. And that can be as simple as removing, the denaturant that one had added to unfold it in the first place. And then at this point, what we do is we cross our fingers and we hope that the protein magically snaps back into its original state. And if it does, then we say, goody, now we get to do biophysics on this protein. And we can follow this process of it reassuming its native state using a panoply of different techniques, such as circular dichroism, but also things like FRET, NMR, anything that's really ever been done as a physical technique in the chemical sciences has been ingeniously adapted to also study this, more complex process of protein folding. And the long story short is that proteins that have been studied heavily in the last 4 or 5 decades generally do have this property that they can relocate their native state upon being unfolded. And the model that is typically used to explain this phenomenon is shown on the right. So this funnel, known as a folding funnel, is basically showing that you have to imagine the y-axis is being energy, and the z-axis is being energy, and the x and the y-axis in this plot sort of represent some coordinate that represents the shape of the protein. And what this funnel architecture is showing you is that it doesn't matter how you unfold the protein. Any unfolded state is going to be able to find its way back down to the native state because that's the free energy minimum. And thermodynamics would dictate that it doesn't really matter what path you use to get there, doesn't really matter how you unfold the protein, because the native state is the most stable. Thermodynamics guarantees that there's always going to be a good way back down, which is, it's sort of reminiscent, in some ways to those games that you have, in malls where you have these funnels and you put a coin on it, the coin goes round and round and round. It doesn't really matter where you put the coin, it's always going to find its way back to the bottom. And that's because thermodynamics dictates that by gravity, the bottom is the free energy minimum. So this is basically that same concept, but now in a much larger, a much higher dimensional space, where instead of there being one coordinate in which gravity causes things to come down, you have many coordinates for all the different amino acids and atoms in the protein. But there's a little bit of a problem with this model, which is that even though every biophysicist knows this, few are willing to say out loud that actually most proteins do not refold if you take them apart. So if we take proteins that are more complex, such as ones that are inside membranes, or such as ones that have many, many different subunits, and you perform this same experiment where you denature the protein and then return it to native conditions, in this situation, more often than not, a protein does not refold into its native state. More often than not, it precipitates, which is not the native state. So in the context of this funnel, where we are always saying that the minimum was the native state, what are we to make of this type of observation? Well, I think if you take the empirical reality at face value, what it's telling you is that maybe it's the precipitates, the aggregated form of the protein, which is in fact the free energy minimum. And if that's true, then the interesting question is, if the precipitate is the minimum, then what did biology do to get us into the native state in the first place? That to me is an interesting question. So our hypothesis, and not just ours, there's a number of, I would say, biophysicists and cellular biologists who are intimately interested in this question. But our hypothesis is that the additional information that is necessary to help proteins fold is encoded in translation. And translation acts as this sort of assembly line that helps this manufacturing process of constructing proteins, and that the way that proteins get constructed on this assembly line helps them navigate this energy landscape and help them find their way to a native state that you would not be able to find if you were only relying on thermodynamics alone. So what are ways in which translation could act as an assembly line? Well, we know, from work about ribosome profiling, which Kimena, one of your colleagues, has worked with one of the world leaders and inventors of this technique, is that we know that certain codons are read faster or slower than others. So what this means is that the choice of codons that you use for particular amino acids amounts to a set of traffic instructions, so that you have fast codons that allow ribosomes to whiz on by, and that could bias you to form certain secondary structures that form more quickly, such as alpha helices, whereas other things such as slow codons or strong secondary structure in the mRNA could have the opposite effect of slowing down ribosomes, causing them to essentially get into traffic jams. And these jams might not only be pathological, they could also have an effect of helping to template the assembly of proteins that are forming in a large multi-subunit assembly. So we know, for instance, in bacteria, that oftentimes if you want to have a multi-subunit protein, the genes that encode those different subunits will be on the same mRNA molecule, which we call an operon. And in this sort of like cartoon that I'm showing you here, that operon could encode spatiotemporal cues to help the nascent chains find each other at specific points in space and time, and thereby help promote the biogenesis of a complex protein. And needless to say, this is a hypothesis that is intimately intertwined with translation. So it's not a type of process that we would expect to happen efficiently if proteins were refolding by themselves in a test tube, which is exactly the experiment that we are normally doing in biophysics. So this is sort of, I think, frames a number of interesting questions that we have. So firstly, how does translation help proteins find their native states? And how does the kinetics of that process allow us to basically go beyond what thermodynamics would make possible? So I'm going to be talking about this in the second half of my talk. But before I do, the first half of the talk is going to actually, in a sense, take a step back. And the step back was really thanks to one of my first graduate students who joined my lab last year, who said, Stephen, you go on and on about how important kinetics are for protein folding, but clearly there are some proteins where thermodynamics is enough. Like if you take them apart, they can snap back to their native state. And the question that my student Philip asked was, which proteins can refold reversibly and which ones can't? And when he first asked that, I said, well, clearly someone has thought about this and there has to be an answer somewhere out there in literature. But there really isn't. And the reason why is because the dogma through which we've been thinking about protein folding has been so dominated by thermodynamics for the past 4 decades that if an experiment ever showed that a protein could not reversibly refold, then it basically wasn't ever published because that was just considered a negative result. So that sort of leaves us with an interesting question that in some ways is very old, but also very new, because really, even though people have qualitatively found that proteins don't follow the rules of not refolding, they haven't really ever been methodically cataloged in that way. So we sought to address this question, and our motivation for this question is we're interested in trying to set limits on what proteins might depend on these co-translational processes to help them fold. Because if they do, then we hypothesize that they might not be able to refold on their own in a test tube. So in order to think about this, I want to help walk you through an experiment that Philip has been carrying out in the last few months. And I want, if you have any questions about this experiment, to raise hands now, because it's definitely my goal that you can, if you don't remember any of the results, that you walk away with what's this approach that we've been developing, because I think it could be an approach that's useful for a number of other questions. So what we have done is that we start with cells, and in our initial experiments, these are E. coli, but there's nothing about the experiment that would require us to use E. coli, so we're interested in doing it on other cells, other organisms as well. And we've lysed those cells under conditions that do a very good job of keeping all proteins intact, so in their native structural form. So it's basically a very gentle mechanical lysis. What we then do is that we divide that sample in half. And to one half, we do nothing to it, and we call that the native sample. To the other half, we unfold it using a chemical denaturant, and then we refold it by removing that chemical denaturant very slowly. And what this does is it generates a refolded sample. But what I really should do is put refolded in quotes, because not all of the proteins would have successfully refolded. Some may have, and that's represented by that alpha helical structure that I'm showing on the left, but other proteins in this process might not have successfully relocated their native state and may have either aggregated or become some protein that is still soluble, but nevertheless misfolded and not in the native structural conformation of that protein. What we next do is we take these two complex mixtures of proteins, so these two lysates, and we structurally probe them using a protease called proteinase K. And proteinase K is an interesting protease because it has virtually zero selectivity at the amino acid level. So it basically will cut at almost any single amino acid with the same efficiency. But proteinase K does have a strong preference, and that is to cut at regions that are unstructured or accessible. So what that means is that if you are a location in a protein that is exposed to solvent and not in a stable secondary structural element, then proteinase K will very efficiently cleave at that site. But the locations that are tucked away into the protein core or in strong secondary structures are, at least for brief periods of time, immune to being cleaved by proteinase K. So this is useful for us because it means that if a protein is identical in the two different samples, meaning that it has the same conformation in the refolded sample as in the native sample, then we would expect proteinase K to cut the protein in the exact same locations and to the exact same extent in those two sample types. But if the protein is misfolded, then that will presumably expose new regions to be susceptible to proteinase K, whilst occluding other regions that might have been accessible in the original native sample. So essentially what proteinase K is doing is it's allowing us to take structural information on these large complex collections of proteins and encoding that into the form of a cut site. What we next do will now look familiar to anyone who's ever done a routine proteomics experiment, which is we cut up with the enzyme trypsin. And trypsin you can think of as being like the foil to proteinase K, because it doesn't care how structured or unstructured you are, but it only cuts after two residues, and that's arginine and lysine. So what I'm showing you here is the pattern of tryptic fragments that will be created from treating the protein with trypsin. But now in orange, what I'm showing you is that some of these tryptic fragments have gotten interrupted because there was a proteinase K site in between two sequential trypsin cut sites. So what that means is that we're going to wind up with a very complex mixture of peptides, some of which are what I would call fully tryptic, which means that the beginning and the end of the fragment were defined by a trypsin cut site. But some of the fragments will be these so-called half tryptics because the tryptic fragment was interrupted by a proteinase K site. And we care a lot about these half triptych peptides because by sequencing them, we're able to infer where did proteinase K cut the sample. And where did proteinase K cut that protein? But most importantly, if we compare the relative abundance of a given half triptych peptide between the two samples, then that allows us to infer whether or not the protein was able to refold or not. Because if a protein could reversibly refold, then it would have the same structure in the two different sample types. It would generate the same proteinase K cut sites, and we would see all the peptides that map to that protein in equal abundance in the two different samples. But if a protein could not reversibly refold, then that will generate novel fragments that would be absent in the original sample. So I want to actually stop here for a second in case anyone wanted to ask any questions just about this experiment. And I'll go out of full screen so that way I can see if people are raising their hands or not. Okay, Yi Xian raised her hand, so I will allow you to talk.
Can you hear me?
Yep, I can hear you.
Okay, so my question is, when you do this assay, isn't it very important to have the correct timing in your unfolding, in the sample that you're doing unfolding, comparing to your native state? How do you define how long you need to wait until you do your proteinase K?
Oh, that is such a good experiment. Yeah, so there are many, many timing steps that are important. So the amount of time that proteins are able to unfold is important. The amount of time you give them to refold is important, and we'll talk about that. The amount of time that you allow it to cut with proteinase K is extremely important because we need to get this kind of like sweet spot condition where you essentially have, you know, sites being cut, but not so many sites that you're basically just destroying proteins, you know, to smithereens. So that ends up being 1 minute. So yeah, timing is really important at all of these steps.
Thank you.
Great question.
I have a quick question. Do you eliminate all of the aggregates? So the things that aggregate, are they just lost, or do you do something with them?
Okay, I'm glad you asked that as well, Will. And actually that comes up in a few slides, so I might just hold that for a second.
Oh, good.
But yeah, you read my mind practically. Okay, I'm gonna go back to screen share unless more hands come up. Okay, great. Here we go. Lovely. Oh, we need to move this to the side. There we go. All right. So before we went and did this experiment on whole proteomes, which was our intention, we first wanted to pilot it on a simple system, namely a single purified protein that has been extensively studied as a model for folding, and that is staphylococcal nuclease. So staphylococcal nuclease is a small, very stable single domain protein of about 200 amino acids that, as you can see by circular dichroism in this upper left-hand plot, appears to refold, meaning that if I take that staph nuclease, take it apart, put it back together, and then compare the CD spectrum to the original protein, they would appear to be indistinguishable. And that's also quantified by looking at the critical mean residual ellipticity at 220 nanometers shown in that bar chart. Now, in our experiment, we did the same thing where we took staph nuclease, either did nothing to it or unfolded it and refolded it, probed it with proteinase K, and then prepared for mass spec and analyzed the peptide fragments. And the way that I'm representing the data from that experiment is with this thing called a volcano plot, which I want to walk you guys through in case not everyone here is used to seeing them. They're very common in proteomics. Essentially, each dot on this plot represents a peptide that we were able to successfully sequence in our experiment. And on the x-axis, the X value for each dot represents some type of effect size, which is basically how much more abundant was that peptide in either the native sample or the refolded sample. And that's plotted on a log2 axis. So things that are around 0 are of similar abundance in the two sample types, whereas things that are much more to the right represent peptides that were much more abundant in the refolded sample, and points that are far to the left were much more abundant in the native sample. And the y-axis represents the statistical test against the null hypothesis that the peptide was present in equal abundance in the same two samples. So basically, things that are very different, should be sort of in the upper right or the upper left-hand corner of the volcano plot, where the red represents some like region that we would ascribe significance to. Importantly, what you see in this volcano plot is almost all the points are really clustered at the bottom. In fact, you can't even see all the points. We have 200 different peptide sequence, but they're almost hard to make out because they're basically all at 0, 0. And what that's telling us is that by our experiment, just like CD, staph nuclease is a refolder, meaning that we can't discern any structural difference between the protein that was initially prepared versus the one that has been unfolded and refolded. So that's kind of nice, a nice sort of sanity check, and basically is telling us something very similar to the CD experiment. But importantly, an experiment that we can do with mass spec that we cannot do with CD is that if we take this same staph nuclease protein that we've unfolded and refolded, and we have that spiked into an E. coli lysate, which of course has 4,300 other proteins in it, we basically get the exact same results, which means that our experiment is very good at looking at these profiles of refolding peptides, even if they're in a very complex background, which is of course one of the main strengths of mass spectrometry. It's very good at analyzing complex mixtures, whereas by contrast, if we had done the same thing by CD, you wouldn't really be able to discern any signal from staph nuclease amidst this complex background of all the other proteins in E. coli lysate. So that was a really nice experiment for us to see. And we repeated this experiment also with RNase H, another common model protein for studies of folding. And the story is very much similar, that it is identical. The native and the refolded state are identical by CD. They're identical by lip mass spec. And that is not changed if the protein is in a complex background. So an interesting sort of experiment that we did with these next, which I wanted to sort of throw out there, is that at the beginning, I said that how do we refold proteins? We basically unfold them of a denaturant and then we get rid of the denaturant. In practice, there's two common ways of getting rid of a denaturant, and that's either you just dilute it so that the denaturant is at a low concentration, or you use dialysis to basically trap the protein inside a little compartment, and the denaturant, being a very small molecule, dialyzes out. So the data that I showed you before was when, oh, Will has a question.
Hey, sorry to interrupt. Is each dot a peptide or?
Yes,
Okay,
each
cool.
dot is a peptide that we have sequenced. And if that is a peptide that is a half triptych, then it means we can map it back down to a location where proteinase K cuts. Because we basically know that this is a site where proteinase K, or where trypsin would not have been able to cut, because it's not an arginine or lysine, and therefore we can infer that that's where a proteinase K cut site happened.
Cool.
But yeah,
That's
each dot represents a peptide.
Thanks.
So what these experiments showed us is that actually RNase H and staphylococcal nuclease, when they are refolded by dialysis, are actually not exactly the same protein anymore. So in particular, RNase H seems to slightly expand, and there are a number of residues that are sort of at the edge of the protein that are now more accessible than in the native protein. And similarly, staph nuclease actually does the opposite. It kind of compacts, and some residues at the end of alpha helices have now become harder to cut in the refolded state. And so this was sort of interesting to us kind of as an aside, because, you know, typically this type of folding problem is always looked at by CD. And by CD, everything, unless it's completely, you know, different, will basically look the same. And what this was sort of showing us is that we have a method that can actually probe these ensembles of proteins very sensitively, and something that everyone would assume that RNase H is refoldable under any condition actually is not true. So this is a protein that's kind of been a poster boy for thermodynamic refolding. And what we're showing here is that it does refold perfectly fine by dilution, but not under dialysis, which is another condition that is commonly used. So it's a little bit of an aside, but basically just to sort of say that we have much more amino acid level resolution and much greater sensitivity to probe subtle differences in protein structure in solution than would be normally possible with other structural techniques. Okay, so now I want to basically really go back to the important question that Will asked a few minutes ago, which is, what about aggregation? So we worried a lot about aggregation, because the problem with our experiment is that if you take a lysate and you unfold it in a denaturant and then you remove that denaturant, then most of us would assume that the main thing that would happen would be that you would just get a lot of aggregation. And the reason why aggregation really concerned us is because you could imagine a situation where one protein, let's call it protein X, aggregates when you try to dilute it out of the denaturant. And another protein, let's call it protein Y, had it been by itself, would have been able to refold. But now, in the presence of this nasty aggregate caused by protein X, it's just going to sort of join that dog pile, right? And then the information that we get would be harder to interpret at the level of individual proteins, because the aggregate would sort of affect the whole entire mixture. And so before we kind of dived into this, we wanted to invest some time to try to see if could we develop any set of conditions that would make it possible to minimize aggregation upon trying to refold an entire lysate. And what this bar chart is showing you is if we basically measure the percent of protein that is in an aggregate relative to the initial starting material, we found that if we did our denaturation using urea and at a slightly elevated pH of 8.5, we could really minimize the amount of aggregation. So it's basically about 1% of the total protein mass, which is kind of remarkable considering, you know, our BCA assay probably has an error bar of, or a background level of about 0.5%. That's what's shown on the left-hand column, which represents a native lysate that was never unfolded at all. And what this chart is showing you is that urea results in significantly less aggregation than if we had used guanidine as our denaturant. And this pH effect is also significant, especially when in combination with urea. As a sort of like follow-up to that, we did a separate experiment in which we compared how much aggregation or how much precipitation we have if we had done a thermal denaturation. And that means we get proteins to unfold by heating them up, and then we get them to refold by slowly cooling them overnight. And sort of consistent with what most would expect, thermal denaturation results in much more protein denaturing or getting aggregated, and that effect can somewhat be mitigated by simply doing your thermal denaturation at a very low concentration, just so that it's harder for proteins to find each other. But the long and short of it is that we felt like we were happy with this high pH urea denaturation for the purposes of studying refolding on lysates, with aggregation being a minimal concern. Okay, so now that we've laid that groundwork, what do the data tell us? So I'm once again showing you a volcano plot. And the volcano plot, in this case, once again, each dot represents a peptide that we've sequenced. The x-axis is an effect size. The y-axis is a test of statistical significance. But now, instead of this being conducted on a single purified protein, it's being conducted on all of the E. coli proteins in the lysate. And what should jump out at you when you look at these data is that you have what looks almost like a trimodal distribution, where there's a very, very large number of peptides that are sitting right at 0. So that means that they are present in equal abundance in the native and the refolded sample, and then a much smaller but non-negligible number of points that are off to the wings at very large and very small values. And what this distribution tells us is that the refoldability question really actually is binary. Like, there are proteins that truly can refold, and as a consequence, they basically look indistinguishable between a native sample and one that has gone through this refolding process. But then there are proteins that generate fragments that are effectively absent from the other sample. So these are cut sites that are unique to the native protein or unique to this refolded protein. And as a consequence, we basically don't detect those peptides at all in the other sample type. And that's what these sort of big lobes off to large positive and large negative values are. But there was a problem that we encountered with these data. This is a little bit more technical, but possibly interesting for people who work with proteomics or sort of large data sets, is that the problem with our experiment was that we were really, of course, interested in these data points that were sort of off to the flanks, because these are the ones that are, different between the two sample types and are therefore, evidence of non-refoldability. But the problem is that the way that these points occur is that you essentially don't detect that point at all in the other sample type, which is concerning because it means that you're making these important claims essentially based on the absence of evidence. And unsurprisingly, when you try to make these strong claims based on the absence of evidence, you get relatively low reproducibility, right? So there'll be certain situations where you just don't observe a peptide in one experiment, but then if you repeat it again, you do see it. And that worried us a lot. So we spent a lot of time trying to develop a new algorithm to basically filter through the raw proteomic data and generate a way to reproducibly measure peptide abundance over about 10 orders of magnitude. So we were very pleased once we were able to sort of get through that reproducibility problem. And with that, we can now sort of ask this question: how many proteins are able to refold and how many can't? Well, our experiment gives us evidence for 1,355 E. coli proteins. 280 of those, we don't really have enough data to make a strong claim about whether or not they refold or not. But of the remaining 1,000 proteins, roughly half of them we find to be a non-refolder, and the other half we find to be refolders. So let me define what I mean by those terms. So we call a protein non-refoldable if there is more than one peptide that maps to that protein that was statistically significantly different between the native and the refolded samples. So just having one for us was not good enough, because we wanted to make sure that we could sort of see evidence for that in at least 2 different locations of the protein. Now, importantly, in this refolding camp, which I'm showing in black, that includes a number of sort of classic famous proteins, such as triose phosphate isomerase, dihydrofolate reductase, and cyclophilin A, which are E. coli proteins that have been studied in vitro and have been found to be refolders. So importantly, in this extremely different experiment that's been done in a very large complex sample, we're able to reproduce the refoldability of certain key famous proteins. But also importantly, and perhaps more interestingly, half of the E. coli proteins seem to be unable to refold. So that led us to next ask, what's the story of these proteins and what can we say about them? So a very simple hypothesis that you might ask, or that you might wonder, is that we might think that big proteins are bad at refolding because they're just kind of big and complicated. And so that's a hypothesis that we can test. We can basically look at what's the distribution of molecular weights of all of the refolding proteins, as well as the molecular weights of all the non-refolding proteins. And lo and behold, there is actually a very significant difference between those two populations, according to the Mann-Whitney test. But that is only significant really just because we have so many data points. It's not a particularly large effect size. And actually, I would argue that it sort of went against my intuition that indeed most proteins really were able to refold even very large ones. So yes, bigger proteins are less likely to refold, but not in a way that is like truly compelling. A much more interesting, I think, result that came out of this was we also then filtered the proteins for which ones were monomeric versus which ones were multimeric. So a monomer is a protein that's composed out of one peptide chain, and these proteins are actually, by and large, very good at refolding. Whereas proteins that consist of multiple different peptide chains, whether or not they're all the same sort of peptide or distinct ones, were generally much worse at refolding. And that to me was a very interesting observation, because if we go back to our model of how proteins fold on the ribosome during translation, we have this sort of picture that if you have a multimeric protein, it in principle has the ability for these different chains to find each other whilst they're still being made on the ribosome on the same mRNA. Whereas in our in vitro experiment, of course, they have no such luxury. Those proteins, once they're unfolded, will have their sort of subunits sort of split apart from each other, which now creates this challenge. They have to find each other, which was not the case when it was folding the first time, because those chains would have been in this kind of microenvironment created by being on the same polysome. So that was, I thought, kind of interesting. Another way that we were able to look at the data was whether or not proteins were better at refolding depending on what their location would be in the cell. So what we found is that ribosome proteins were really bad at refolding, and that of course makes sense, because for ribosome proteins to refold, they generally need this rRNA, this long ribosomal RNA molecule to act as a template, which is of course absent in our refolding experiments. Somewhat intriguingly, we found that membrane proteins were better at refolding, which actually is sort of surprising, but in a way it makes sense, because one needs to remember that the true transmembrane proteins are actually just absent in our experiment, because when we lyse cells, those are just going to aggregate out, and we clarify those away. So the membrane proteins that we're looking at here are ones which have a large cytosolic component, but are somehow tethered to the membrane by perhaps a single pass. And actually, it kind of makes sense that proteins like that would be good at refolding, because in the biogenesis of those proteins, they would have to be unfolded in order to be passaged through the translocon. So in a sense, these membrane proteins that have like a single pass membrane but a large cytosolic component, it's that cytosolic component that we're looking at in this experiment. And it makes sense that those would be good at refolding, because in many cases, the cell had to unfold them in the first place in order to be able to translocate them across the membrane. So that was an initially puzzling result, but actually makes sense if you put your cell biologist hat on for a moment. Another thing that we could do is that we came in with the hypothesis that maybe certain proteins that came from certain fold classes or certain fold types would be better at refolding. And for the most part, that is not true. So if you look at all these different classes of fold types, it turns out that pretty much all the major folds have evidence for non-refolders and refolders at proportions that roughly echo the overall population. And so what this basically suggests to us is that non-refoldability is not likely to be some ancient trait that proteins had at the origin of life and which were then inherited by all proteins from that clade. But rather, what the results are suggestive of is that early proteins were probably all refolders, because in early life, we didn't really have a sophisticated translation machinery to make co-translational folding possible. And then over the course of time, proteins accumulated mutations that made them non-refoldable and therefore dependent on the translational machinery. But those mutations were all neutral because, of course, they happened in the context of a cell that had a ribosome and therefore was able to perform efficient co-translational folding. So we basically don't see that there's certain classes of proteins or certain fold types that are intrinsically non-refoldable or not. They basically occur throughout all different fold types. But I would say probably the most interesting and the most sort of compelling results that we found is there was a very, very strong correlation between the number of domains that a protein had and its ability to refold. So long story short, the more domains a protein has, the worse and worse it gets at refolding. And this, again, is very consistent with a lot of current thinking on co-translational folding, because it's believed that the ribosome is good at pausing translation between successive domains. And so what that means is that the protein has an opportunity to essentially fold up one domain before it has to worry about the next domain coming out. And so in a sense, this lack of refoldability with proteins that have more domains, but not necessarily are larger, just that they have more domains, is consistent with this idea that the traffic instructions, if you will, during translation play an important role in the assembly of these types of proteins. And finally, I'm gonna sort of stop showing these plots soon, but I think I'll sort of end with this one. So the result that pretty much, surprised me more than most of the others was that we found a very, very strong signal for essential proteins to be non-refoldable. So essential in this context means that you can knock them out in E. coli and you can still have a viable strain. This has been extensively studied in E. coli thanks to the CHIO collection. So basically, we know for all 4,300 E. coli proteins whether or not you can knock them out, and that results in a viable or non-viable strain. And what we found is that proteins that are essential are actually very, very frequently non-refoldable. Now, our explanation for this is that naturally essential proteins include things like the ribosome and like DNA polymerase and RNA polymerase. So these are proteins that are, of course, very complex. And so essential proteins tend to be complex. And so there does seem to be probably, a confounding variable in that sense that, these large multi-subunit proteins that perform these kind of complex manipulations for the central dogma tend to maybe non-refoldable simply because they're big and intricate. But in any case, this was one of the more interesting sort of results from our data. And it's again kind of almost consistent with the fact that we know that the last universal common ancestor about 3.8 billion years ago did have a very sophisticated translational apparatus. And so basically what this is saying is that life as we know it basically depends on the ability to use the ribosome to co-translationally fold proteins and not be solely reliant on thermodynamics to get them to assemble. So this actually goes back to one of the early questions that came up in the talk, which was, what about the kinetics of things? So I mentioned very explicitly that we're very precise in how much time we allow proteinase K to cut, because we sort of need to be in this sweet spot where we're getting, you know, a good number of half triptychs, but not so many that we're just ripping up the proteins to shreds, and then we wouldn't be able to sequence it anymore. So that's something that we optimized, and that's just simply a technical parameter of the experiment. But a sort of more scientifically interesting question is, what if we gave the proteins more time to refold? So to give you a sense of how this experiment works, we have our native protein or our native lysate. We unfold and concentrate it down with urea. We then dilute it into a native buffer after giving it a long period of time to unfold. And then we wait distinct periods of time post dilution into native buffer. So either 1 minute, 2 minutes, all the way up to overnight. And then at those distinct time points, we then probe with proteinase K. And therefore, we're able to get a set of fragments that are indicative of the refoldability of the proteome after distinct periods of time following their dilution from the denaturant. And what the data tell us is that for the most part, proteins that are able to refold are generally able to do that within the first minute. Again, that's consistent. Most model studies of protein folding show that proteins in some cases can refold in milliseconds. So especially small single domain proteins. So we basically have 580 proteins that are refolding in a minute. If you give more time, that number does go up. At the 2-hour time point, that number has gone from 580 to 620. So there's a small number of proteins that need that extra time. And all of the sort of data I showed you up to this point was on that 2-hour time point, which is kind of a sweet spot for refolding. It turns out if you wait overnight, it actually goes down a bit, but that's probably simply because certain degradation just happened. The sample kind of went a little bad over that time. But yeah, 2 hours, you seem to have proteins that have done the most refolding without just non-specific sort of cleavage, from proteases inside the lysate. So we did do that kinetic experiment as suggested. So to wrap things up, in a nutshell, I hope that I've been able to convince you that this limited proteolysis mass spec technique is a very sensitive way to detect protein structural differences. We can get information at the amino acid level by honing in on exactly what amino acid proteinase K cut at. And we can also do this experiment not just on single proteins, but on the whole proteomes, so that we can probe refoldability on not just single proteins at a time. I hope that I've been able to convince you that non-refoldability is pervasive. So there are many proteins that if you take them apart with a denaturant and then ask them to sort of refine their native state, they fail to do so. And that's evidenced by these peptides that we only detect in the native or the refolded sample. And that by looking at that data collectively, we could pick out a number of very strong signals suggesting that in general, multimeric proteins, multi-domain proteins, and essential proteins are especially bad at refolding. So we are still relatively new into this project, and we're excited about the results that we have, but there's lots more that we want to do. So one experiment that I was hoping that I'd be able to share with you today, but we just ran out of time, unfortunately due to the shutdown that happened 2 weeks ago on Hopkins, was we wanted to now ask, what happens if we throw chaperones into the mix? So what happens if we take proteins that we know are not able to refold if you simply take them apart and put them back together, but now we take them apart and throw in chaperones upon their refolding experiment? We'd be interested in knowing which proteins now get rescued and are able to successfully refold once we give them a little bit of extra help in the form of E. coli chaperones. And we're also, of course, interested in what this effect looks like in other organisms. Our hypothesis entering into this is that we imagine that the more sort of complex the organism is, the more proteins will not be able to refold. Of course, we do not know that, and we want to do the experiment to find out. So in the interest of time, I'm probably just going to not talk about the second half, which is fine. We can always talk more because, of course, I'm local to Johns Hopkins. And so if this stuff interested you, I'm happy to talk to you more. And I will just zip off to my acknowledgement slide to say that, obviously starting off a new lab, you're extremely indebted to people that sort of get things off the ground. And I feel very lucky that I got some great graduate students last year in my first cohort. So everything that I spoke to you today was due to Philip. Annalise has been developing some really exciting methods that use cross-linking mass spectrometry to really try to dig in deep and get structural information about what co-translational folding intermediates look like as they're coming off the ribosome. And that uses cross-linking approaches in order to trap structural information about nascent proteins into cross-links that we can then sequence on the mass spectrometer and use in order to create structural models of these folding intermediates. And of course, I also want to thank the funding agencies that have helped support us in these early stages, as well as our lovely mass spectrometer, which when she's having a good day, she makes us very happy and gives us lots of data. And when she's not having a good day, makes us sometimes pull our hair out. But we've had enough good days that we can share with you the data that I got to talk about in this last hour. So thank you very much. Sorry for maybe going a little bit over, and I will turn off screen shares so I can see your questions.
Okay, thanks so much. I'm going to clap. I don't know if you can hear me, but thank you for a wonderful presentation. And yeah, if people have questions, I see there's already a hand up, so I'll let Stephen manage that. All
right, so we are going to start. I guess I might start with Michelle Biederman because Yishan asked a great question already, but then Yishan, you are next up.
All right, can you hear me?
I can hear you.
All right, I guess since it seems like you were doing these whole proteomes and you were talking about the chaperones, were you able to, or you have any plans to try to do the proteome just without the chaperones in there?
So you mean like deplete the ones that are endogenously present?
Yeah, I would, because I would assume that there's a mix of, if you're doing the whole proteome, that there's chaperones present.
Yeah, no, that's a fantastic question. So I am not overly worried about that, and for two reasons. So one is because when we go from cells to lysates, we basically dilute the endogenous chaperones by over 1,000-fold, but also more importantly, we dilute the ATP concentration to below 1,000-fold, such that the ATP in the lysate is well below the KDs to the chaperones that depend on it. And E. coli has one ATP independent chaperone that's trigger factor, so that could possibly still be operating in our lysates. but all of the other like sort of critical chaperones like GROEL, GROES, DNA, JKE would really not be active because of the ATP concentration. So I'm pretty confident that what we are showing you here is refolding in the absence of chaperones, and to then sort of do the refolding with the help of chaperones, we need to basically resupply it so that it's at a concentration that is relevant. So basically about 1,000, basically just express it, purify it, and then re-add it back in at a concentration of about micromolar, as well as adding sufficient ATP. But yeah, great question. I'm going to now allow Yixian to talk.
Okay, now you can hear me, right?
Yep.
One of my questions, I have two, but both are fairly short.
Great.
One of my questions is about the folding reaction. So you're doing it in the lysate.
Yes.
So what's your sense about, when you have a protein lysate and then you denature them, whether you could have interference from various other proteins also in the solution, or you think they're essentially, having enough space and doing their own thing, not interfered by other denatured proteins? What's your sense?
Yeah, so I think like the way in which we've thought about that is that essentially it boils down to concentration, right?
Right.
And you could imagine that if you had a really, really dilute lysate, that you would minimize the possibility of proteins sort of like forming accidental interactions in the lysate. But the problem with diluting it, I guess indefinitely, so I guess like to the homeopathic levels, is that then if proteins form, some important complex, so let's say it's like an oligomeric protein, then you also make it ridiculously hard for those to reform too, right? And then a protein that doesn't reform, some native interaction is then going to, of course, appear to be a non-refolder. So in practice, we have this, 0.23 mg per mil, concentration, which is in a sense arbitrary, but it basically is this nice condition that allowed us to minimize the aggregation while not diluting it so far that it would become impossible for the multimeric proteins to reassemble. But I think that you raise a good point, because in a sense, if you're doing something in a lysate, the concentration practically by definition becomes this kind of like arbitrary factor. Like obviously in the cell, everything is at a concentration that's roughly 1,000 times higher than what's in our experiment. And so that's basically just kind of one of the limitations, I think, of this in vitro experiment.
Yeah, well, so my second question is, it's a technical quick question. You said you had to get a mass spec to do all of these experiments. Is your mass spec a standard mass spec, or it's just something specific?
Yeah, thanks for asking that. So the one that I guess made an appearance on our acknowledgement slide is called a Q-exactive HFX, which is a specific model for essentially an Orbitrap. So the whole field of proteomics practically uses Orbitraps now for about the last 10, 15 years. So we're all very much like at the behest of Thermo Fisher, unfortunately. But they really have made a superior product, and the Orbitrap kind of balances accuracy with speed. And the accuracy is important because in order to tell different peptides apart that are very similar, you need this like part per million sort of mass accuracy. And the speed is important because our samples are very complex. And so, when you take an E. coli lysate and chop it up and then send it on liquid an LC, you basically have, maybe 100 different, distinct species that are eluting off the column at any given time. So what that means is that we basically need to be able to get those mass spectra very quickly so we can try to see as many things as possible before, something else comes off the column. So yeah, it's a somewhat specialized instrument, but it's standard for proteomics.
All right. Thank you very much, Steve.
You're
This
very
was a
welcome.
wonderful talk. It's wonderful to have you as our colleague.
Oh, thank you. That's very kind. And I'm very, very glad that this has worked out.
Yes.
All right, I'm gonna try to field 2 from Steve DeLuca and then Robert Levis. So Steve DeLuca, you are up.
All right. So your experiment showing that or your findings showing that the multi-domain proteins tend to be enriched in the non-folders, and that's suggesting that really like the effective concentrations of these like unfolded domains really screws up folding.
Yeah.
And the solution would be like to pause ribosomes in between domains. That would definitely help. But I was wondering, like, if you have an RNA with multiple ribosomes on it making multiple polypeptides, they could, interfere with each other through the same type of, effective concentration mechanism. So did you look to see if like initiation, whereas like, translation and initiation, or even transcription in an E. coli was like slower for these like multi-domain proteins that are also hard to fold, or did you look for a correlation with
So that's
more upstream things?
So that is a very good question. So, so I have to say, so I want to say this right. So there was a really, it was a good paper, but I don't agree with it in Nature Structure and Molecular Biology by Sarah Teichmann's lab. And they were interested in this question. They basically were wondering, for homodimeric proteins, is the interface between the two different, between the subunits, does it tend to be on the N-terminal or the C-terminal part of the protein? And they're basically what they showed is that there is an enrichment for the interacting region of a homodimeric protein to be on the C-terminal part. And they thought that that was interesting because in their model, it basically gives a protein an opportunity to fold into the monomer before it assembles into the dimer. So basically, like, the region that you expose to form the dimer doesn't get made until like you're near the end. And so basically it gives you a chance for like a protein to sort of like come off and then fold into a monomer, and then that monomer to then go and find its partner and form a dimer. And they basically, in the whole paper, go on and on about how there's like really like statistically significant effect of a p-value by 10 to the -8. But then if you actually look in the supplementary information, it turns out that like this enrichment is only like a 10% enrichment. So like, again, it's one of those like, hilarious things where it's statistically significant because you have like 4,000 examples. But the truth is that it's roughly 50-50. I mean, so what I would basically say is that I think that when it comes to assembly of multi-subunit proteins, there are probably going to be situations where, E. coli wants to just make the protein, have it form a monomer, and then have that monomer find its partner in the cytosol. And I'm sure there are also situations where the assembly itself is co-translational. And you can make the hypothesis that that would be more often when the sort of interaction interface is N-terminal. Which is basically slightly less than 50% of all multimeric proteins in E. coli. So I think that you probably have like, both situations happening. And I guess, as you can guess, I'm personally more interested in this situation where the assembly is co-translational, but there are probably situations where it's not. All right, great question. I'm next going to allow Robert Levis to talk.
Stephen, you characterized the proteins as either foldable or non-foldable, but I was wondering, particularly for multi-domain proteins, whether part of the protein might fold and part remain unfolded.
Okay, so that's a great question. And there's really two, I think, ways of interpreting that question, which are both really important. So one thing is, since we're getting information that we can resolve down to individual amino acids, like we know where protein K cut, like why did we have to basically then it's like sort of then zoom back out to like the protein level? Like, couldn't we have then taken these like, you know, these sort of distinct sites and then sort of do more of a structural analysis on them and map them to specific domains or specific secondary structural elements? And the answer is yes, we just haven't done it yet. My expertise in structural bioinformatics is, you know, sort of middling at best. So I probably would be interested in kind of publishing a story that is just about these kind of like descriptive statistics about different types of proteins that refold and don't, and then really in a sort of separate follow-up kind of dive into the kind of like other structural features or domain types that, you know, sort of tend to harbor those types of non-refolding features. So it's a great idea, and we just haven't done it yet. The other way of maybe thinking about your question is, are there proteins out there that refold some percent of the time, right? So, you know, where basically you unfold it, and then with some percent probability, it basically manages to navigate its way back to the native state. But some other just population, so, you know, still the exact same sequence, but just, you know, because of the stochasticity of, you know, traversing this energy landscape, some basically get bifurcated into some region that is non-refolding. So I think that the best way to sort of think about that is that if that were true, then we would expect there to be a large number of situations where if you quantify the relative abundance of a peptide between the native and the refolded sample, what you would see is kind of something that's, you know, like, you know, twofold or fourfold or, you know, sixfold, let's say. But going back to that kind of like original histogram kind of earlier on in the talk that kind of showed that trimodal distribution, where you have like a large population that's basically at 1-to-1, and then these other large populations, you know, that are basically at the detection limit of the instrument, you know, like 100 or 1,000 to 1. It does actually, in my mind, suggest that you really have this property kind of like hard coded into the landscape, that it either goes back or can't. And I'm sure there probably are some situations where you have that kind of, you know, mixed population of things that non-refold and or things that refold, and then some population that doesn't, but that does not seem to be a standard or a common behavior.
I don't know if you can still hear me. I had a technical question, how you do the partial proteinase K digestion, how do you inactivate it without messing up other proteins?
Oh gosh, what a great question. So we have some folding enthusiasts. I'm gonna go back to screen share, and I'm gonna go back to that slide. I can definitely answer that. So essentially what we do is that we do proteinase K very rapidly. So we have a tube that has our aliquot of proteinase K in it. We then take the lysate and sort of like rapidly pipette it in vortex, wait for a distinct period of time. Then in order to rapidly inactivate proteinase K, that actually ends up being very subtle, because proteinase K, of course, is itself a very hardy protein. So what we do is that we have a bath of mineral oil that has basically been heated to 180 degrees and Celsius. And then we take our sample of protein that's been incubating with proteinase K for 1 minute. And then we basically dunk it into like super hot oil, which basically causes everything to unfold, including proteinase K. But at that point, it doesn't actually matter, because we've already sort of encoded all the structural information into cut sites. So as long as we, even if we unfold everything, as long as we unfold proteinase K as well, we're not going to get any more cutting. And then, so that sample that's been like globally unfolded by sort of basically boiling in really hot mineral oil, we then transfer that back into solid urea, which is convenient because dissolving urea cools your sample down because the dissolution of urea is endothermic, and simultaneously basically keeps proteinase K turned off because it unfolds at 8 molar urea. Then what we do is we dilute it to 2 molar urea and then add trypsin. And this works because proteinase K ends up not being reversibly refoldable, but trypsin is basically still folded at 2 molar urea. So very detailed, but maybe more than you cared to know, but that's how we do it. Okay, one new message. Oh, okay, I think that might be all in terms of questions. Oh, we have maybe Michelle again and Fred. So maybe we'll do Michelle again and then Fred.
Hello again, sorry. Yeah, I don't know if there's a really basic question or not, but is there a difference in the number of disulfide bonds per protein that is able to fold or refold?
We should look at that. We haven't done that yet.
I was like, classic Amundsen. I don't even know if this is too basic or not.
No, I mean, I'm actually like sort of, I'm actually sort of, I feel bad I haven't done it yet, but we should totally do that. Yep, that's a great question. We should do it. Haven't done it yet.
Cool, thanks.
And Fred, I think you might have our last question.
Along the lines of looking at multi-protein complexes, I'm wondering if you've looked to see, whether it looks like there's 1 or 2 core components that seem to fold, whereas, the rest don't. Trying to see about cores versus accessories.
Yes, we should do that, and we haven't done that. Gosh, you guys have so many great questions, or great ideas, I should say. Yeah, no, the way that we have annotated things as multi-subunit or not has been to simply use the annotation that is on the EcoPsych, which is the E. coli encyclopedia. It does, of course, get to be into a somewhat dicey business because it does get to be a sort of dicey business because, of course, there are proteins that are not, that can exist as both, right? Where they're stable as a monomer, but then they form a multimer in response to some signal. And of course, proteins like that should follow the same trend as the monomers, right? Because if it can exist in some stable constitutive state as monomeric, then it shouldn't require the assembly to be able to fold. So we did at least do that. So EcoPsych does a good job of distinguishing between what it calls subunit composition and biological assembly. So we used biological assembly, which would not classify something as a multimer if that was formed more as a regulatory event. But yeah, beyond that, we haven't really gone into excessive detail. And so I think that you're absolutely right that for like large assemblies, there's an interesting question of like, if something is constitutive, it forms like a core, we would expect it to sort of have this behavior. Whereas if it's like something that's peripheral that comes on and off in response to a signal, that it shouldn't. I guess we're just relying really on the annotation data that what we have called a multimeric protein is something that is basically only exists in that multimeric state. Although I think that once you start thinking, about the cell biology, you can start to see pretty quickly that this becomes a slightly arbitrary dividing line, which might explain why like that, like chi-square was not as high as some of the other ones, because, it might be that it's more complicated than the way that we have divided it up, but certainly a very good question.
Yeah, so I guess maybe just like staff nuclease, if you find a couple, very well-studied complexes, that might give you a little bit more confidence in it.
Yeah, that's a good idea. I think that the thing is that when it comes to like the model proteins in refolding, like all the model proteins are these small single domain monomeric proteins. And that's sort of, I think, in a sense, like kind of, what we hope like the impact of the work is that, there's kind of been this like, picture, of like what is a, model protein for folding. And it's basically always been this like, small single domain monomeric. And it's basically, I joke, it's like these proteins are very privileged. We need to basically democratize folding to these kind of other cases.
Thank you.
Okay, I'm gonna just step in, and I think we should probably wrap it up to give Stephen a break. I know he's been on Zoom for about 4 hours now.
This is our new life. I guess we're all going to get used to it sooner or later.
Yeah, but that was a wonderful seminar. Thank you for kicking things off. Yeah, that was great, and I think a bunch of us will probably follow up with you with questions. That was yeah, really nice talk.
Thank you, Will.
And thanks everyone else for, yeah, thanks everyone for joining in.
I guess before everyone leaves, I know that Steve, we spoke about meeting after the seminar. Is that still okay with you? Do we know if Steve?
I think he sent an email to you, follow up.
Okay, I'll just email him. Okay, all right, take care everyone. Stay safe.
Awesome talk.